drug dosing and the best ways to do it, and kind of the best scientifically and optimal ways to do that. So that's what we're going to do. I'm going to share my screen, and we'll get started. All right. And can everybody see that?
Yes, I can see it.
All right. If you have any questions, you can put your little hand up in Zoom, and if I see it, I'll certainly stop. Let's make this informal as possible, like it was in person, and have a good discussion about this stuff. Okay. So why do we interspecies scale? And there's a lot of reasons why we do this, and the classic one is to determine the starting dose for human trials. Is you have safety data in 1 or 2 species on a pre-IND application, and you're starting to develop a drug, and you got to come up with a way to say what are we gonna do first to put into people. So a lot of that is to determine starting doses for human trials. And the other one is, if you have rodent data, how do you determine your doses for your second species safety trial as you're in preparing an IND package, and you need safety and toxicity in 2 species. And this is a colloquium that I put together, and I included residents from both veterinary medicine and human medicine. And a lot of times we use this data to estimate doses in veterinary medicine. And I say this a lot, I teach veterinary pharmacology and toxicology. A lot of times the human is the preclinical species when you're doing veterinary medicine. The data you have is from humans, and you're using a human drug off label in dogs. How the heck do you figure out how to dose them? And so you have to start with some type of scaling, because it's not like there was a registration trial done in dogs for that drug. So your dosing estimation has to come from somewhere. And I'm gonna kind of focus on this today, because we do a lot of this. The concept of bench to bedside and then back to bench to optimize use, and it takes us really 20 years to figure out how to use a good drug optimally in all the kind of clinical situations where it's going to be used. And so those preclinical studies of approved agents and agents that are being used for other things are important. And the reality is, and I'll show some data, is that if you use good dosing in your preclinical species, you can get data that is very much more reflective of what you're going to see in humans. So a way to optimize your preclinical studies, so you're not just giving mice doses that are completely irrelevant to what we know we can get in people. And so I'll talk about this in cases where we have doses of drugs in both people and rodents, and why you should think about that when you're doing your rodent studies in these these bedside back to bench situations. So there's a lot of reasons, and to go from rodents to dogs, dogs to rodents, dogs to humans, humans to dogs. And that's why I put the arrows both ways. There's a lot of reasons to want to go between species and think about how we're using these drugs and what we can learn from them to optimize their utilization. Now, a lot of people, and I've heard this hundreds of times, seems like when we'll be talking about scaling, they'll say, well, just use allometry. And I go, oh, okay. And allometry can be useful, and it's biological scaling, and it generally measures changes in organisms in relation to body size. And some things have been established to be related to body size, and one of the primary ones is cardiac output. And here's a study from 1968 that shows that ventricular volumes and body weight in mammals scales to a power of the 0.75. And so you can go from rats to cows and horses, and you can actually extend this up to elephants and even larger things. And you can scale cardiac output by this power to the 0.75. And the reason that's useful in pharmacology, right, is that a lot of our drug elimination is related to cardiac output. And so In the kidneys, it's clearly related to glomerular filtration, as well as delivery to the proximal tubule for active secretion, and also reabsorption as you dilute the local area and you drive reabsorption for drugs that can be reabsorbed. So renal blood flow is a function of cardiac output, and thus cardiac output can play a primary role in drugs that are renally eliminated. It also plays an important role in hepatic clearance for high extraction drugs. So drugs that are efficiently extracted and either metabolized or excreted into the bile by the liver, when you don't have any kind of barrier to that because the liver is very effective at it, it really comes down to how fast drug can be delivered to the liver for that clearance. And at those cases, for high extraction drugs, hepatic clearance approaches hepatic blood flow. And since that hepatic blood flow is related to cardiac output, again, you get cardiac output is playing a role. And so there are some drugs where allometric scaling with body weight works quite well. And it turns out they're mostly, a lot of them are very water soluble antibiotics. And here's some work by Jim Revere, who's a veterinary pharmacologist and has done a lot of this kind of allometric scaling with these drugs, showed that gentamicin is eliminated, entirely by renal elimination. And when you can look at the half-life of gentamicin, and it scales to body weight. So in those cases, you can use allometry to calculate out doses, and it has been used to calculate out doses as you go from all kinds of different species in veterinary medicine. Now, he did a whole bunch of studies of this, and only about a quarter of veterinary drugs you could actually make predictions with allometry, and these were almost all antibiotics, and again, water soluble antibiotics. So this really fits that model that you have to have clearance and elimination correlate to cardiac output and to these functions dependent on cardiac output to get allometry to really work. Okay, and so I think that's important to note. Allometry can help you get in a range, but there's going to be a lot of other players that are involved. And the reason that you get more variability is that you get species differences in metabolism. The P450s are very different in various species. And in veterinary medicine, as soon as you get into the cats, then all of a sudden everything goes wacky because they have deficiencies in conjugating enzymes as well as transporters. And this is really related to the fact that now you have a species and a group, the felines in general, who are hypercarnivores. They don't really eat plants, and so they lost a lot of the metabolic capacity associated with that. And I always say that if everything you eat had a liver to take care of it before you ate it, then you really don't have to worry about having those enzymes. So there's no evolutionary pressure to keep a lot of those phenolic conjugating enzymes, and so they lost some of the major glucuronidation pathways. Species differences in transporter proteins, organic anion, organic cation, and physiologic differences that you don't really think about: urinary pH, gastrointestinal pH, biliary flow, blood flow to certain organs can be different. And so there's a lot of things that can kind of really alter this ability to just scale using allometry. And what I'm going to talk about for the rest of this discussion is that empirical data will really help you get the rest of the way. So experimental data and using data that's already out there in a lot of cases can allow you to make some very, very nice estimations and calculations going back across various species. So sometimes you can just go, is the data there? And it's there. The PK data may be there for multiple species, and you can work that out. Okay, so we talked about body weight. What about body surface area? And there's a lot of body surface area has a tendency to be a little better correlate with clearance than just body weight. And although body surface area is a function of body weight, it's also a function of some other factors. And there are tables that have been worked out, and these are all utilized heavily in calculation of NOEL and low-L calculations. An example here, a safe and effective dose of drug X was determined to be 50 mg per kg in mice. How would you convert that to a human equivalent dose? there are based on body surface areas. You can convert from mgs to kg back to mgs per meter squared, and this would calculate out to be about 150 milligrams per meter squared in a human, or about 4.1 mgs per kg. So that's an interesting thing, just thinking about it right up front, that a 50 mg per kg dose in a mouse, if you just scaled it based on body surface area, is less than a tenth of that in a human in terms of mg per kg. So, you know, those doses are going to get a lot smaller when you start scaling that way. And this method is based, you know, again, on that concept that drug clearance scales with body surface area. And that's not always true. All right. That's not always true, but it can give you a ballpark area. And I'll give you an example with drug now. So let's go from the human dose to a dose in the dog. And so let's, you know, in veterinary medicine, like I said, sometimes the human's a preclinical species. If we go back and we look, and the standard human dose of doxorubicin is about 60 milligrams per meter squared. And if you just do a BSA conversion of this, this comes out to be about almost 3 mg per kg. That's actually 3 times what the dog dose is. It's 1 mg per kg or 30 mg per meter squared, usually in the dog, which is interesting because that 30 mg per meter squared dose is actually closer to what we give kids. That's more of the pediatric dose for doxorubicin is in that. And I use this example because I knew it would fail, because I knew that dogs didn't scale that way. But what gets really interesting is that if you take and look at the pharmacokinetics, you look at the plasma levels of doxorubicin in human and dogs, and this is from some human data and dog data that I had with the humans dosed at 60 mg per meter squared and the dogs dosed at 30 milligrams per meter squared, those curves are almost on top of each other. And so it's not that dogs are more sensitive or anything like that to dox. It's that they distribute and metabolize it differently. And when you give them the safely determined dose in dogs, it looks just like the safe determined dose in humans in terms of overall exposure, as shown by the area under the curve. And so, you know, the same exposure gives the same response. So the biology is not different. It's just the distribution, metabolism, and elimination of this drug such that the dose that you give them becomes a little bit different. So that clearance is different, and there's no underlying biology that's really different. So, and I think this is a really good example of where the PK data, once you have it, can help really give you an idea of how to do these cross-species doses, because now you know exposure. And I think that's an important thing, and is that we're not really talking about dosing. We're going to go to exposure when we start talking about pharmacokinetics, because what pharmacokinetics does, right, is it gives us a relationship between dose and exposure. And once we know exposure, and we know that exposure is generally proportional to response, which is kind of a general kind of rule in pharmacology. Once you know that exposure, then you can start doing the things you want to do in terms of comparing PK across species and matching that dose with exposure. Now, there's a couple caveats in here. One is that the underlying biology is the same across species, such that the same exposure does result in the same response. And that may not necessarily be true with a lot of targeted agents because of the underlying mutational spectrum associated with the tumor and the driving response. If, I mean, not in mice implanted with human tumors, because what you're treating is the human tumor, but if you're treating, you know, a mouse tumor or a tumor from, you know, some type of syngeneic or other species, you know, and a lot of the PK-PD relationships that we've kind of worked out over the years and we rely on come from antibiotics. And in that case, the PK is the important part because the target is the same in all species. It's the bacteria. And so it's that, you know, their response is going to be the same regardless of what species they're in, based on exposure. So in that case, the underlying biology will be the same, but we have to just make sure that the underlying biology is the same. This is also data-based, so data in both species must be available for comparison. And I hate to say it, sometimes you can trust some data more than others. So you have to be a little bit selective in terms of who did the study and what you're going to do. And we've run into problems with that in the past. And then you need to understand actually what you're trying to compare. Are we comparing Cmaxs? Are we comparing Cmins? Are we comparing AUCs? And so how does this drug work? Do we have a minimal concentration we're trying to maintain? Do we need to talk about exposure, which is AUC, which is both time and concentration, or is some max level that we need to get to? And for a lot of drugs, that's there. For a lot of the older drugs, for some of the newer ones, it's not. And for a lot of targeted agents, we kind of go under the assumption that you need to stay above a threshold for a certain amount of time, which is probably right in terms of inhibiting the target. But if that's not established, it may be, you may be able to do that in tissue culture. Just show if you can, if you pulse them for pulse cells for X amount of time, can you block proliferation? Do you need to go longer? What do you need to induce cell death? And so you can ask those questions in terms of your exposure and response potentially in other systems. But there are some caveats that you need to think about as you're thinking about the pharmacology of these agents. And it's interesting, and I put this in here because this is some data I've been actually working on lately in our own studies. And is the underlying biology the same? And so I actually made this figure just a couple of weeks ago. So I was like, I'm going to put this in because it shows it is. And this is actually getting to a lot of what I'm going to talk about next month in terms of in vitro, in vivo exposures and making sure that they're relevant to the pharmacology. Here's some canine and human osteosarcoma cell lines and some human breasts sprinkled in there, looking at inhibition of proliferation by hydroxychloroquine. And these are some studies we're doing looking at autophagy inhibition. And the point here is that the underlying biology looks about the same, is that you have, most of the cells, show, well, all of the ones here show inhibition of proliferation between 5 and 18 micromolar. And you get some that are sensitive and some that are resistant, more or less sensitive within the human, within the dog, within breast, so it goes across histotypes, and you get, kind of this general response that's seen in all species. So you don't have all the human on one side and all the dog on the other side. And the added bonus, and this is going to get again to my lecture next month, at least 50% inhibition is shown at doses that are achievable in humans, or concentrations that are achievable in the plasma humans, which is what I show in those lines. So just something to think about. You got to make sure that you can actually get that much drug in what you're treating if you think you need to inhibit it at that level. But I think the underlying biology, if you can show it's the same, these cross species comparisons become a lot more powerful. Okay, so we talked about human equivalent dosing, and so thinking about what level you can actually get in the population you're trying to treat versus what you can get into a mouse. And this is I think an incredibly important paper that came out in 2012. Effectively what this paper shows is that, and it's really shown in the panel on the right with A and B, is that if you treat at a dose that is relevant to humans, or like a human equivalent dose, you actually get a correlation between the overall response rate seen in clinical trials. And I've sat in a lot of Gordon conferences over the years where people have said, mouse models are not predictive. And I've heard some very good scientists, Pete Houghton's the primary one, tell them that's because you don't know how to use them. And I think that this data really starts to get to that, and that you need to use doses that you can actually achieve in the people that or the animals that you're treating in the mouse to make the mouse model predictive. And that if you overdose, or if you do MTD in mice, because we don't really care how sick they get, you can kill and you can get a lot of anti-tumor response that you'll never see in a population where you're limited by dose-limiting toxicities. And so, and this study, which came out of Genentech, I think is quite good at showing that. I put the paper up there if you want to go back and look at it. But the responses measured in mouse models using simulations, and in this case they did kind of simulations because they had curves, but they had the data at clinically relevant exposures correlated with clinical response rates. And that's shown in B, where you get a very nice significant correlation across a series of drugs, including targeted and cytotoxic agents, that when you use levels you can get in people, you could actually predict the overall response rate that you're going to see once you get to a population. So proper use of mice is required for you to get good data out of them going forward. And this gets to why these interspecies scaling in the bedside back to bench situation is important to do. If you're going to take it back to the bench and you have the data from the bedside, use that data to make your studies more predictive. Okay. And this really was something that became clear to us a while ago. And this is when we started working with vandetanib, which back then was known as ZD6474, which was a dual ERB1 VEGFR2 inhibitor that AstraZeneca was pretty keen on as being like a super ARESA to treat lung cancer initially. And here's some data with humans with vandetanib, and you know, the thing that really stuck out to me when we started doing these studies, and this was, oh God, probably, you know, 2001, 2002, was that this drug had a really long half-life in people, 120 to 200 hours. And so when we started trying to relate that to mice, and we knew that this would be dosed daily in people, we had to you know, kind of take into account the fact that in people it was going to accumulate, and that's shown here as here's a single dose PK day 1. You can see it goes up and then it just kind of stays flat because it doesn't, you know, elimination's really long. It's not going to go down to way over here. But at day 29, when you stay on it, now you've accumulated almost tenfold. And with that half-life, you can actually calculate an accumulation factor. If you know the half-life in the species, and it's 1/1 minus e to the negative KEL times the dosing interval. So it's how fast it's cleared, and the dosing interval play an important part, as you would guess. And we could calculate that out, and it comes to be about 7.1. And so we could take this data and we could multiply it basically by 7 and kind of work out what the Cmax and the AUC for this should be at steady state at each of these doses. And then say, okay, if now we know what the levels are going to be in the humans treated with this drug and the recommended dosing going forward, and this drug's been approved for treatment of RET positive thyroid tumors, which was not even on the table when AstraZeneca was thinking about these studies a long time ago, because they didn't even know about these RET thyroid tumors. But in the original studies, the safe drug was 300 mg per 300 milligrams per day. So right here. And so we knew that information. So we knew how much you could get in a human, and we did some PK studies in mice, and the half-life in mice was 24 hours. And so we said, okay, that's not 120, but we can use the same accumulation factor, and a half-life of 24 hours gives you an accumulation factor actually of 2 if you're dosing daily. And we could actually estimate what the mouse should be at steady state too. And we estimated that, and then we actually measured it, and our guess and our measures came out pretty good. So the math worked. And we could do that same thing and figure out these responses, what your Cmax and what your AUC, these are, oh, I guess I put the same curve in here twice. One of these should be Cmax, one of these should be AUC. But you can see the exposure of the drug with dose we could do with the mouse. And the key here is then taking the human data and the mouse data and overlapping them. And here's the Cmax at 10 mg per kg for the mouse put on the human curve, and the AUCs at 10, 25, and 50, 10, 25, and 50. And what you can look at here is what the level is in the human at steady state at 300. So that tells you that the dose in the mouse to match the Cmax in people is between 10 and 25. And you can actually calculate that out exactly. I think it's like 18 or 19 mg per kg in the mouse. And if you look at AUC, and this is because the half-life is so much longer in the human, that dose gets a lot smaller. Now it's less than 10. So if you're matching AUCs, now you need to do about 10 mg per kg. Okay, about 9. And so what this allowed us to do, and I remember when I had these conversations with the folks at AstraZeneca when we did these studies, and it really came down to very interesting, because in lung cancer, this stuff didn't work. And so when I said you should probably be dosing mice in these studies at around 10 to 12 mg per kg, they said that doesn't work in the mice? And my response was, well, your drug doesn't work in humans either. And so you really need to, I mean, and it turns out there was some subsequent modeling that was very nicely done by Erica Bradshaw that showed why this was, why it didn't work in these EGFR driven tumors, but it did work in the RET tumors that it wound up getting approved on as Caprelsa. So the PK works when you actually use it and extrapolate this. And in this case, you know, if we're going to do these studies, if you're going to do drug combination studies with vandeatinib, which is approved now, you know, I would say 10 to 12 mg per kg is where you want to be in the mouse, even though you can give them up to 100 mg per kg and they're fine. That, you know, you can't get there in people. And so doing the studies in mice is going to give you a false sense of activity that's, you know, not going to be very helpful. Okay, so this is an example, and there's other ones where we've done similar things, where when you have the PK data in the mouse and you have the PK data in the human, you can do these types of direct comparisons with taking into account accumulation factors for daily dosing to get to a point where you can do this type of human equivalent dosing that's based purely on empiricism. So this is just using the data and comparing it and coming out with the doses based on those comparisons. Okay, and I think it's a nice example of where this worked. And like I said, there's subsequent data to show why this drug worked with RET-driven tumors, but it didn't work with EGFR-driven tumors. Okay, so if you remember, I said metabolism is one of those things that is hard to take into account. And so can you, you know, because since it's variable, can you just use metabolism to predict the pharmacokinetics of drugs across species? And a great example where you might want to try that is with cyclophosphamide for a couple reasons. Number one, cyclophosphamide is metabolized in species, and that's its major route of clearance. The other thing is that the metabolite is actually the active form. And so this 4-hydroxycyclophosphamide needs to be formed for this drug to be active. It then breaks down into aldephosphimide and into the putative active agent, phosphoramide mustard, which binds to DNA and kills the cells, and acrolein, which is thought to be partially responsible for some of the bladder toxicity associated with this agent. But the point here is that this P450 step is critical to its metabolism, activation, as well as clearance. And so can you do that? And the reality is that when you look at the kinetics of cyclophosphamide metabolism, and this is just some V versus S curves, classic Michaelis-Menten. This is just 3 different human microsomal preparations to show that there's variability, 2 different cat, 3 different mouse, and 4 different dogs. The point is that you see this variability, and look at the human, look at the concentrations you have to use to get the same type of metabolism you can get it in the dog at 1/100th that. And it turns out that the human is really, really inefficient at metabolizing cyclophosphamide, and dogs are really, really good. And in fact, at one time back in the 90s, there was talk of using, and we've known this for a while, but of using the dog genes as a transgene to get more cyclophosphamide activation in people. Kind of a bad idea because you can just give more in people. But and anyway, so we've known this for a while, and the mouse is also pretty good. The cat's kind of in the middle, and so the dog's the best, followed by the mouse, followed by the cat, and the human's way, way behind when you look at what we would predict for clearance based on comparison between Vmax and Km. And the cool thing about this is that you can actually reflect this in activation of cyclophosphamide to killing. And so this just shows a bioassay where we used each of these microsomes to generate active drug. And again, what you see, the dog is the best at the lowest concentrations. You can kill the most breast cancer cells. That's what we're using as our target cell. And humans, you got to get the much higher concentrations before you can start killing these cells, because you're not generating the reactive metabolite as well. Okay, so and you know, this was important for us to show because this really got down to the biology of what was going on. Okay, so what does that have to do with pharmacokinetics? Well, what we then did was we took this and we built out a pretty simple physiologic model, because you have to add blood flow, because blood flow to the human, the cat, the mouse, and the dog liver are a little bit different. So you need to get things going in the right way. And when we did this kind of simple semi-physiologic model with metabolism of the drug right here, we could do a pretty good job predicting the pharmacokinetics of cyclophosphamide in dogs, cats, mouse, and in humans over here, using data from our 3 to 4 microsomal preps. So we could predict roughly the half-life, or at least get within a pretty fair assessment of the differences in humans. And again, in humans, look at the time in minutes. It's way out here, 1,000, 1,500 minutes, where we're a lot shorter in mice, dogs, and cats. And so when you had that metabolic data, you could do some relatively simple modeling to predict the pharmacokinetics and the half-life of this drug. And work out, one would predict that based on the formation of the 4-hydroxycyclophosphamide, you could actually predict the different toxicity doses, and that is actually true as well. You can predict how much of the reactive metabolites generated in each species, and that correlates to how much you can actually give them before you see toxicity, and the dose in humans is much higher. Okay, and so what this tells us is that when you take into account the metabolism, and the metabolism is driving that pharmacokinetic parameter across species, you can do species-specific parameters. And you also need to include a little bit of allometry with blood flows, because those are critical to having in there as well. So again, more data, you can make better predictions. Okay. The last example I'm going to use is putting everything together, and this would be what we generally consider the gold standard for interspecies scaling. And this is using a little bit of allometry, a lot of experimental data, and really playing out the physiological differences and the structural differences with blood flow in each species, as well as using metabolic species-specific parameters to model the pharmacokinetics of a drug. And this uses blood flows, organ volumes, metabolism, transport, secretion, all kinds of stuff. And I'm going to look at this with first with a drug that's got a lot of attention for all the wrong reasons in the last year and a half, and that's hydroxychloroquine. We were interested in looking at to see if we could predict the pharmacokinetics of hydroxychloroquine, mostly because of the fact that it sequesters in lysosomes. And if you get pH changes within a tumor, this is where we really got interested. You can see differential uptake and distribution of this compound. So we needed to describe it overall in the body before we could add the tumor compartment. So we started doing some modeling with hydroxychloroquine. And basically what this stuff does is it accumulates in lysosomes because they're very acidic, and you trap these diprotonated forms here because they got 2 charges. Now they can't cross membranes. So the lower the pH, the more you accumulate in there. And the tissue uptake of these things is blood flow and this lysosomal surface area and the flux between the lysosome and the cytosol, which has permeability differences that are associated with those pH differences. So anyway, I'm not going to go into that very much, but it's just physiochemical properties of the drug are critical, as well as the physiology and some of this biochemistry. And so now, and I put this up here because here are some of those biochemical measurements we talked about with cyclophosphamide. Here's some of these allometric methods, things that we talked about with allometry, blood flows, organ volumes. These are all kind of mix of allometry and biochemistry and some of this weird physiochemistry of hydroxychloroquine, where it has these pH dependent stickiness in various organelles because of its charge. And so the point here is we built this model, and you can look in here in blood and liver. Here's blood, here's liver. We can put this together, and it's always important to point out that these lines are not curve fit. These are pure simulations from this model. So we could generate these curves with or without that data. And the data is there just to show it's pretty accurate. We can predict the pharmacokinetics in the mouse when we put these things together in a structure that is appropriate. And we can actually predict different tissue levels, gut, kidney, things we had measured. And then the key to these is you can, if you have you might need some metabolic data, which you can get from microsomally, like we did with cyclophosphamide. And then you just scale these things based on known physiologic scaled parameters, like how much blood flow to the human. There's books and all kinds of stuff that have these values. So all you have to do is change the allometric scaling, right? You can change it to a human. And when we do that, we can actually predict what happens in the human as well. So here's plasma levels from 3 different studies that we can predict pretty good. Our prediction errors in 20 to 30%. That's actually not bad because you're predicting these things, de novo. And if you get within 2-fold, you're doing pretty good. We can predict urinary elimination. And so this scaling was just, we didn't have any human data except data we got from the literature, and just scaled it and saw how it fit. And we were able to predict hydroxychloroquine disposition in these human patients. And if we took it and we went and looked at kind of steady state level, and that's the thing about this drug, it has a long half-life and accumulates, you can see that our predictions, now our simulations and what we saw in trials were quite good. And this is from 3 different trials using HCQ in combination with cancer chemotherapy agents. And so we got the HCQ levels, and this was of our interest in this population. And you could see that we could predict quite nicely what the steady state plasma levels, these plasma steady state levels, were at various doses, using this model. So this is really slick because now we can go back and say, okay, and like I showed with the HCQ response and the osteosarcoma, I know what the human plasma levels are. Well, actually, this is whole blood, not plasma, but whole blood levels are. So we know how much of this drug we can get these tumor cells exposed to through the blood. And so we can actually now start translating that to better studies in vitro, in tissue culture. And again, that's going to be the basis of what I'm going to talk about next month. But this kind of combination of allometry and biochemistry and physiochemistry is the gold standard, where you start taking everything into account, and now we can make really nice predictions from mouse, where we built the model, to humans. We've gone to dogs, we can do that as well. We've done that in the past with other agents. We could predict, cats if we wanted to. You can just, even cows, you can start changing the parameters allometrically with proper biochemistry. And now we're taking into account a lot of those factors that I talked about and getting better predictions. Now, these cross species PBPK, doxorubicin, we've done a fair amount of that. We've predicted drug-drug interactions in various species. We've predicted what would happen in dogs that have transporter polymorphisms with no data, pure simulation, and went back. So these really start giving you the ability to go cross species and actually have kind of different situations you can do. Docetaxel, we've gone cross species. Lapatinib, we've gone from mouse to human. Vandiatinib, we talked a little bit about. Hydroxychloroquine, like I just showed, and our cyclophosphamide data, which is a lot more of a kind of simple physiologic model, but it's there. Okay. All right. So hopefully what I was able to get across was that there's a lot of ways to do interspecies scaling, and that a lot of times you need to do it because you need to have a starting point, and that's what we do when we calculate out starting doses in humans, and we use NOELs and AOL and NOALs in critters, and we try to work back to species in humans. And in that case, when you don't have human data, you have to do that. I mean, you just have to figure out a way to do your best scaling, and then you throw a safety factor in there and work your way up. Allometry is generally limited to cases where drug PK is driven to blood flow. Database comparisons, when you have data in the species of interest, you can just start doing these right across comparisons, and that's just pure empirical. Just cross your curves and see where they cross, and you can then figure out the doses to use. Those are really powerful because you already have the supporting data. In vitro metabolic data can be used for prediction in combination with some physiologic factors. And, people have tried to predict overall clearance and intrinsic clearance using metabolic parameters. And there are softwares that are written to do this. Sometimes they work, sometimes they don't. Sometimes the allometry and some other things are really critical. And then, the gold standard really involves all of these things. You got allometric components, physiochemical properties, biochemical components, and that this might be overkill in a lot of situations, depending on the question being asked. And so I would never say you have to do these models for everything to do interspecies scaling, but they open up avenues and allow you to do things that you may not be able to do in special populations or across some other species. So, data-based, the more information you have, the way you use that information is critical to doing good interspecies scaling. Okay. All right. And like I said, using PK data for better in vitro and vivo drug dosing study designs, what I'm going to do next week. So I'm going to take some of this data as well as some other stuff, talk about it in study design. And like I said, what I really want to do is to get this as an interactive thing that we're doing. And, hopefully other people want to talk and can present stuff and really get, some nice educational things going on cancer pharmacology. I'll stop my sharing and I'll gladly take any questions that people have.
Dan, this is Mike Veneris. I have a quick question for you. You showed mostly data on kind of traditional chemotherapy, which requires kind of a certain set of enzymes to break down, metabolize, et cetera. I'm curious if the same principles apply for like small molecules like tyrosine kinase inhibitors and JAK inhibitors, and how you think about that. I guess it's still blood flow issue, GFR kind of issue, or how do you think about that? And of course, this is relevant to the emails I've been sending you about trying to do PK dosing and JAK inhibitor mice. But anyway.
Yeah, so we've done this same thing with lapatinib. Vandiatinib's a TKI, and so there's been a lot of this done with TKIs, and they certainly can fit into this. Just like, even some of the cytotoxic I talked about, where I think it gets a little more difficult is well, it's not difficult. It's just, the players are different with biologics because the clearance systems aren't necessarily as clear, and distributions based on a lot of other factors like body composition and stuff like that. So antibody drugs, I think, become a little more difficult. But yeah, with TKIs and stuff, all these things are directly applicable. And like I said, we did it with lapatinib and vandiatinib. And I actually did a very similar thing with Iressa years and years ago and calculated out doses when we were doing some stuff with Gale. So yeah, we have that data for gefitinib as well.
Great, thanks a lot.
The other part that's cool about that, and I don't know if, I'm not sure if Natalie is on, but there's some really cool ways to do imaging correlates. We did that with vandetanib and some pediatric leukemia models. And Natalie did a great job of looking at both imaging and then the metabolomic correlates of that with MR spec. And I don't know if you want to go into that at all, Dan, but it's really cool. And it gets you a very, as early as, like 12 and 24 hours after treatment, you can see metabolomic correlative changes and imaging changes, which is also super cool, that can then help guide some of the PK markings.
That's an awesome idea. I'm going to email Natalie as soon as I get off done with this and say, hey, why don't you give one of these on imaging based PD endpoints for drug response? Yeah. I mean, the probes have been tough to do that with, but if you have the right probe, I remember Teresa Troiani doing some. You remember we looked at some transient changes in angiogenesis to tumors, and she could see some quick changes. They didn't correlate to response all that well, but I remember back in the day when we were doing that stuff. Yeah, that's a great idea, Leah, and that stuff is really cool because you can do it in real time.
Well, and she's got really cool shared instrumentation grants, and also she has much better magnets for animals than we have for humans. It's really quite impressive.
Yeah. Yeah, that's awesome. Any other questions? I know some came up in the chat. I saw. Let me see. So that's a great question. What are the differences between cancer pharmacology and other clinical disease kinetics? The main one from experimental is, cancer and HIV is the only two diseases where we put the drugs we're developing in our patients before they go into healthy volunteers. And it's a little bit different because we walk a fine line between toxicity and efficacy, and our efficacy endpoints are really hard to measure sometimes. So there's been, I think all phase 1 studies in humans have a PK component, right? Certainly for cancer. And so is it different? We can learn things from other disease types, but I think there is a difference in the cancer pharmacology drug development pipeline. And Leah, you might be able to speak to that better.
I think we can always do better, right?
Yeah.
But I think that's, the principle that I always try to think about is, even if the clinical trial is quote negative, meaning it doesn't reach a set statistical endpoint, these correlative kinds of studies can, first of all, maybe tell us why. And second of all, can build upon, how we should refine future trials and go back to the other side of it and think about, what should the different question we want to ask be, and how do we revise the next trial or the next correlative studies to try to target that better.
Yeah. And I think inherently in cancer pharmacology studies, you're always thinking about combinations from day one. So that's always, gonna be in there. What's the polypharmacology gonna look like in this population? All right.
Dan.
I know this was recorded. It'll be put up. Feedback. Like I said, I started this, and I'm just trying to get something going where we have a good kind of discussion panel about pharmacology. It's a little bit selfish to try to get more utilization of people thinking about the drug development and shared resource, and make my kind of profile, and people know me a little more on the Anschutz campus, since it's been a while since I've been there. It's been over 10 years now, so people, those of you that don't know, I was on the faculty in School of Pharmacy and there all the time at one time, and I think it's good to still have a presence, and this is one way to do that. So and with that, everybody have a great Thursday.
Thanks, Dan.
All right, bye.
Thank you.